It is a great pleasure to introduce today Luis A. Ortiz Rodriguez. Luis received his Bachelor of Science in Biology from the University of Puerto Rico, Humacao's campus, where he did undergraduate research studying the photosensitized reaction of phenothiazine derivatives and their interaction with albumins, under the supervision of Professor Rolando Oyola Martin. Currently, Luis is a scholar of the Northeast Ohio Alliance for Graduate Education and the Professorate. He is a recipient of the 2019 Dimitru and George D. Matescu Student Citizen Award and a 2020 Graduate Teaching Award. During his spare time, Luis likes to work out. He's an avid runner and spend time with his wife, Deliris, and with his dog, Miranda. The title of Luis' presentation will be Thionated Organic Compounds as Potent Heavy Atom-Free Photodynamic Therapy Agent.
Okay, so thank you, Dr. Crespo, for that kind introduction. Today I want to talk about some of the research that I've been doing the last 4 years in the Crespo group. And in order to do that, I would like to start by talking and giving some background about skin cancers, but more specifically about non-melanoma skin cancers. Annually, more than 3 million Americans are affected with non-melanoma skin cancers. And non-melanoma skin cancers can be subdivided into basal cell carcinomas and squamous cell carcinomas, which are these malignancies that I'm showing down here. And just to give you some statistics, between 1976 and 2010, the overall incidence of basal cell carcinomas increased by 145%, while it increased by 263% for squamous cell carcinomas over the same period of time. As you can see here, I'm also showing a third skin malignancy called actinic keratosis. And actinic keratosis is a common precancerous skin condition caused by DNA damage in the keratinocytes, which is a skin cell of the epidermis, due to chronic exposure to sunlight. I want to highlight that actinic keratosis is a major public health concern because of the high prevalence and substantial financial impact. For example, in 2004, actinic keratosis prevalence was estimated at almost 40 million in the United States, with an estimated annual cost of $1.2 billion in healthcare related to actinic keratosis. And actinic keratosis can eventually develop into basal cell carcinomas or squamous cell carcinomas. It's very important to mention that solar radiation is the major environmental factor in the development of basal cell carcinomas and squamous cell carcinomas. DNA damage is directly implicated in the initiation of these cancers, in particular the formation of the cyclobutane pyrimidine dimer, which is this photo product that I'm showing here to the left, and the 6,4 pyrimidine pyrimidone photo product, which is this other photo product that I'm showing here to the right. And in our bodies, we have a mechanism that is called the nucleotide excision repair mechanism that is usually in charge of removing these UV-induced DNA lesions. However, this nucleotide excision repair mechanism is not 100% efficient, and that's why some people get skin cancer. There's also a medical condition called xeroderma pigmentosum, in which the nucleotide excision repair mechanism is disabled by mutations. And then if you have this medical condition, then you are not able to remove any of the UV-induced DNA lesions, making these people, they have a higher probability to get skin cancers. As you may remember from last week's talks by Sean Hain, he was talking about the compound methyl 2-pyrimidone, which is this compound that I'm showing here in red. As you can see, this compound is a chromophore within the 6,4 pyrimidine pyrimidone photoproduct. Okay, and as Sean said last week, this compound upon photoactivation can populate a long-lived reactive triplet state. This occurs with a triplet yield of 80%, and this gives So now, since now you're in the triple manifold, you can carry photosensitization reactions or carry photooxidation of DNA. So that's why this 6,4 photoproduct can act as a Trojan horse of DNA damage. That's what I'm showing here, this chromophore overlay with these Trojans on here. So now not only you can form these CPDs or 6,4, which they are mutagenic and carcinogenic, now you can also have this 6,4 photoproduct as a Trojan horse of DNA damage. As we know, there are different treatments to treat cancers and skin cancers. Some of these treatments are radiotherapy, cryotherapy, chemotherapy, and surgery. We know that they can be painful. The cosmetological outcome of these treatments is not good. And they can be pretty invasive. One alternative for these treatments is photodynamic therapy. And the way that photodynamic therapy works is that you have a photosensitizer, which I'm showing here in green. That's a molecule. And with that photosensitizer, you can treat the malignant cell or tissue. The cell will incorporate the photosensitizer, and then you can photoactivate with light that photosensitizer, and that photoactivation will lead to cytotoxic photoreactions that will lead to cell death. Okay, but more specifically, what happens upon photoactivation of the photosensitizer? The best way to depict the different pathways that could occur upon photoactivation is by using a Jablonski diagram. And last week, Naisha Caldero and Sean Hay discussed in detail the Jablonski diagram, but I want to talk about it just to highlight those pathways that are most relevant to the type of research that I'm doing. So as I'm showing here, you can photoactivate the photosensitizer. By doing that, you will populate an excited singlet state by internal conversion and vibrational relaxation. You can populate a lower energy singlet state. From there, you can do internal conversion back to the ground state or emit a photon as fluorescence. But as I'm showing here, I have a pathway connecting the singlet and triplet manifold, core intersystem crossing. This is very important because now I'm connecting the singlet and triplet manifold, and triplet states are usually longer lived than singlet states, and that enable or increases the probability or photosensitization reactions such as type 1 or hydrogen electron transfer, or type 2 photosensitization reactions in which you transfer energy in the lowest energy triplet state to molecular oxygen to sensitize the generation of singlet oxygen. Why is this important? Because singlet oxygen is a highly reactive oxygen species that is well known to oxidize different components within cells. For example, the mitochondria, leading to cell death. Okay, so that's why many photodynamic therapy agents target singlet oxygen generation. After saying that, I would like to give you some of the pros and cons of current photodynamic therapy. Okay, this take into consideration what is available today. Okay, so some of the pros is an effective alternative to surgery. It's a highly localized treatment because you only shine light in those areas where you have the malignancy. It's rapid and non-invasive. It's an outpatient procedure and therefore it's less costly, and it's less cosmetically damaging than the other alternatives that I mentioned. But some of the cons is that photodynamic therapy has a limited ability to target malignant cells. Usually, the mechanism of action is oxygen dependent because you usually target type 2 photosensitization reactions. And this is an issue because in many cases, the tumors, the oxygen concentration in tumors is very low to none. If your molecule depends on single oxygen generation and there's no oxygen, then the molecule won't be effective. Slow clearance from the body, inefficient topical delivery due to molecular size, and there's pain associated with photoactivation. So if we want to develop better photodynamic therapy agents or photosensitizers so that photodynamic therapy can reach its maximum potential, then we have to know those ideal characteristics that an ideal photosensitizer should have. So this ideal photosensitizer should be easy to synthesize and store. It should have high molar absorption in the desired therapeutic window. It should have high triplet yields or low cytotoxicity in the dark, high singlet oxygen quantum yield, and it should be cost effective. Okay, you don't want to develop a treatment or a photosensitizer that then it will be too expensive to make. Which brings me to the first generation of heavy atom-free thionated photosensitizers. As I was talking before, I was talking about basal cells and squamous cell carcinomas. Squamous cells and basal cells are usually are found in skin 40 to 60 microns deep into our skin, and this is important because if you are looking for a PDT agent to treat these malignancies, you need a molecule that you will be able to excite with radiation that can go that deep into tissue. And we have this group of molecules here, the sulfur substituted nucleobases or thiobases. And we learned last week from Nijkas and Schoenstock that in the case of pyrimidines, key positions to control the photostability and or photoreactivity of pyrimidines derivatives are positions 2R4, while in the purine derivative are position 2 and 6. So strategically, in the CRESPO group, we decided to replace oxygen in carbonyls by sulfurs forming thiocarbonyls, and you can see that we have different combinations as I'm showing here. And by doing that, you can redshift the absorption spectrum to the UVA region. Note that 4-thiothymidine is basically a thymidine where we are replacing the oxygen in the carbonyl in the 4th position by a sulfur. Okay, I'm showing here in black the absorption spectrum of CAF-TO DNA, and you can observe the characteristic absorbance at 260, which is common of canonical DNA. So now you can see that these thiobases can be selectively excited without exciting canonical DNA, and that's due to the substitution of oxygen by sulfur. But now I want to talk more about 4-thiothymidine, which is the most widely studied thiobase derivative today. 4-thiothymidine exhibits non-dark cytotoxicity when you treat cancer cells with this molecule. You can selectively excite 4-thiothymidine. I showed that it absorbs in the UVA region while canonical DNA absorbs in the UVC. It exhibits a synergistic toxicity when 4-thiothymidine is combined with UVA radiation, and it's largely independent of the p53 tumor suppressor status. And this is important because the p53 tumor suppressor is a sequence-specific DNA binding protein that is frequently mutated in human cancers and controls the expression of many genes in response to diverse stress, for example, UV radiation. So, of course, you want your therapy to be independent of the p53 tumor suppressor status. So, another characteristic that I think that is very important, I want to highlight, is the selective metabolization of 4-thiothymidine into DNA. Okay, 4-thiothymidine is selectively incorporated into DNA because this process occurs with thymidine kinase, and thymidine kinase is an enzyme that gets upregulated in cells that are actively replicating, cells that are in the S phase, and we know that this is a characteristic of cancer cells. Okay, so now not only you can selectively excite 4-thiothymidine without exciting canonical DNA, now you can selectively incorporate this molecule in cancer cells, mostly in cancer cells. Okay, and you can see that here where I'm showing the cells survival of normal cells versus cancer cells, and then showing that when you combine 4-thiothymidine with UVA radiation in cancer cells, the cell survival is significantly less than when you are using normal cells, and that's due to the proliferation rate of cancer cells versus normal cells. Okay, so but we wanted to study exactly what happens upon photoactivation of these molecules. Okay, and in order to do that, we use broadband transient absorption spectroscopy, which is the main technique in our group. It's a pump probe technique, and we can measure processes happening from the femtosecond to the microsecond timescales. And This is important because we can obtain relevant information on how the excited states absorption spectra are changing over time, and we can observe how population in the excited states is changing, okay? And by using this powerful technique, we were able to show that 4-thiothymidine, when it's a monomer and it's not incorporated into DNA, acts as a type 2 photosensitizer, generating singlet oxygen. So upon photoactivation of the molecule, you have ultra-fast intersystem crossing to a triple manifold within 240 femtoseconds, and you generate singlet oxygen. But when 4-thiothymidine is incorporated into DNA, the preferred mechanism is an 2+2 photocycle addition reaction. Why is this important? As I mentioned before, in many cases, these tumors are oxygen deficient, okay? And now this molecule not only can sensitize the generation of singlet oxygen, it can also do a 2+2 photocycle addition reaction, which is independent of oxygen. On top of that, note that the 6,4 photoproduct, as I showed before, have this pyrimidine chromophore inside that now can act as a Trojan horse of DNA damage, okay? I'm very happy to say that we got to publish these results in Nature Communication, and this work got highlighted by the Nature Communications editor, and in other news media such as the Daily, Eureka Alert, Medical Express, and Science magazine. But we are aware of the limitations that the first generation of heavy atom-free photosensitizers have, and that's the spectral window where they absorb, okay? Usually, photodynamic therapy scientists or people researching potential photosensitizers to be used for photodynamic therapy target the therapeutic window of 600 to 800 nanometers, okay, for obvious reasons. Now, if your molecule absorbs in that region, you should be able to treat malignancies that are significantly deeper into tissue, while with these thiobases, you can only use them for topical PDT applications. Photofrin, which is this molecule that I'm showing down here, is an FDA-approved photodynamic therapy agent that is currently used for non-small cell lung carcinomas and esophageal cancer. Usually, the oxygenation status in these cancers is good, so that's why they target type 2 photosensitization reaction, and the singlet oxygen quantum yield of this molecule is 68%. One effort in the literature to increase the singlet oxygen quantum yield of these compounds is using metallation, okay? So using metal, so you can see that the photofrin or the porphyrin sodium is a porphyrin derivative. So now scientists are using metals, and they are incorporating metals in the porphyrin chromophore to see if they can increase the singlet oxygen quantum yield and the triplet yield of these molecules, okay? But I want to highlight that this molecule is not FDA approved. I'm just showing an example of what scientists are currently doing to increase that singlet oxygen quantum yield. So they are using metals such as indium, gallium, palladium, among others, to do that. But you can see that the singlet oxygen is not that different, 68 versus 70%. And additionally, these porphyrin derivatives they are difficult to synthesize, they are expensive to make, and they exhibit an increased DAR cytotoxicity, which leads me to the second generation of heavy atom-free thionated photosensitizers. So I'm showing here what are the second generation of heavy atom-free photosensitizers, and I'm showing here that only by doing a one-step chemical modification where you have these biocompatible organic compounds that have carbonyls available, you can, by thionation, and this is fairly easy to do, you just have to put in a reflux these compounds with Lawson reagent in a reflux with toluene, you can generate the thionated versions of these biocompatible compounds. And by doing that, you can redshift the absorption spectrum of these compounds, and you can see that I'm going to focus in these compounds that I have here on top. they absorb well within the therapeutic window of 600 to 800 nanometers, okay? And by using broadband femtosecond transient absorption spectroscopy, we were able to show that is in the thio bases, these molecules exhibit ultra-fast inter-system crossing, but now they have singlet oxygen quantum yields of 81%. Remember that the FDA approved have a singlet oxygen quantum yield of 68%, okay? And this, the other compound, Thionated Nile Red, has a singlet oxygen quantum yield higher than 99% and also exhibits high triplet yields and ultra-fast inter-system crossing. I'm very pleased to say that these results, we published these results in the Journal of the American Chemical Society in JACS this month, February. Of course, I'm very proud of that work. And the overall conclusions of this work is that the photochemical properties exhibited by these heavy atom-free thionated photosensitizers are due to a straightforward and versatile oxygen by sulfur substitution of carbonyl compounds, carbonyl groups. Thionation is a convenient and general strategy to redshift the absorption spectrum, is increase the density of states, and the spin orbit couplings between the singlet and triplet manifolds of organic compounds, ultimately enabling ultra-fast inter-system crossing, high triplet yields, and effective generation of singlet oxygen. And now, I want to acknowledge the Case Western Chemistry Department, Dr. Carlos Crespo, for being an excellent mentor, group members, but more specifically Chong Hain, because he has helped me a lot, and we have had very useful discussions about research, collaborators, such as Dr. Han Chao in Rice University in Houston, the National Science Foundation for funding, and the NSF EIGE and NOAA EIGE programs. I'll be happy to take any questions.
Okay, let's thank Luis for an excellent presentation. I have now the floor is also open for questions, so please feel free to unmute yourself and ask questions or put it in the chat, and I'm pretty sure that Luis will follow up. We have about 5 minutes.
Hi, Luis. I have a question.
Yeah.
That was a very good presentation. I enjoyed it. So, comparing your compound, the sulfur substitution that you did, as compared to the one they used the metals such as palladium and got a singlet oxygen quantum yield, which obviously your compound using sulfur, you got close to about 99%. I just wanted to know what really made the sulfur work better, the sulfur substitution. What makes it work better in order for you to achieve that high quantum yield as compared to the other molecules or compound or heavy free atom molecules that the others they used? What makes the sulfur a better replacement?
Thank you for your question, Chris. So one of the reasons that I think that these heavy atom-free photosensitizers are better than those methylated porphyrin derivatives that I show, I think that one of the reasons is, so when you use metals, yes, you have a heavy atom effect, and then you increase the spin orbit coupling between the singlet and triple manifolds, but you might be also increasing the spin orbit coupling between the triplet and the ground state, which is a single state, which can also, of course, increase the rate of decay of that triplet state back to the ground state. The sensitization of singlet oxygen is a diffusion control process. Of course, you need that lifetime of that triple state to be long enough to be able to sensitize the generation of single oxygen. So with thionation, we see that we increase the density of states. So the rate, so by an approximated version of the Fermi golden rule, we know that the rate of intersystem crossing is proportional to the product of the spin-orbit coupling and the weighted density of states. With thionation, we get an increased density of states, and also the spin-orbit couplings increases relative to that of the carbonyl compounds. However, the spin-orbit coupling is not as big as the spin-orbit coupling exhibited by these methylated compounds. So I think that, yes, you increase the spin-orbit coupling, but you increase the spin-orbit coupling that much that also the spin-orbit coupling with the ground state is increasing too much, and then that triple state is decaying too fast, and then you cannot sensitize a single oxygen. So I think that that's one of the reasons of why these thionated compounds are better for this type of application.
Thank you.
Yeah.
Okay, we have time for one more question, if there is any. Okay, I guess there is no more questions for Luis. I'm sure that he will be more than happy and eager to answer any question that you have by email or later on. So please feel free to do so.